Assalamualaikum warahmatullahi wabarakatuh. My name is Siti Nusriyani binti Hussein with metric number 206079. Today, I'll be performing an experiment on alcoholic fermentation in yeast. Fermentation is a process whereby yeast are converting sugars into ethanol, carbon dioxide, and other metabolic byproducts in the absence of oxygen, that is giving the chemical composition and sensorial properties to the fermented food. In the alcoholic fermentation, glucose undergoes the process of glycolysis, forming two molecules of pyruvate. Hence, a net profit of 2 ATP was produced and 2 NAD+ were reduced to form NADH and H+. When oxygen is not present, pyruvate from the glycolysis process will undergo alcoholic fermentation. Two molecules of pyruvate are broken down into 2-acetaldehyde, giving off 2 molecules of carbon dioxide. Then, 2 molecules of NADH will reduce the 2 acetaldehyde molecules into 2 molecules of ethanol. The objective of this experiment is to observe the fermentation activity of yeast and to determine the rate of fermentation in different types of carbohydrate. The materials needed for this experiment are 4 empty bottles, yeast, spoon, balloons, lactose, cornstarch, table sugar, and warm water. Firstly, wear lab coats and safety goggles, and then label all the empty bottles from 1 to 4. Bottles 1 to 3 will have yeast, carbohydrates, and water, while bottle 4 will have only yeast and water with no carbohydrates. Next, fill each of the bottles from 1 to 4 with warm water. And then, add 4 teaspoons of carbohydrates, which is sugar, lactose, and starch into each bottles. I use milk as a lactose and cornstarch as a starch. For bottle 4, there will be no carbohydrates added into these bottles because this will be the control sample. And then, add 1 spatula of dry yeast into each bottle. Mix it by putting hand or thumb over the top of the bottles and gentle shaking. Cover the opening of each bottle with a balloon to seal the end of the bottles. Lastly, observe the bottles and record the observation for every 5 minutes for 25 minutes. Based on the result, the carbohydrate that is added into each bottle will act as an energy supply for the yeast to undergo the fermentation process. The rate of fermentation can be determined by the size of balloon expansion for each sample. The balloon expansion indicates that there is a carbon dioxide release from the fermentation process which is trapped inside the balloons.